I'm started within the nanotechnology. So nanotechnology is the term used to describe the manipulate of matter having a dimensions at least between the 1 to 100 nanometers. This is the case says the US National Nanotechnology Initiative. The different scientific fields that build nanotechnologies such as interface technology, biological science, molecular semiconductor physics, and macrofabrications. Nanotechnology may produce a wide range of novel materials and has a wide range of users in a variety of fields, including energy productions, electronics, biomaterials, and nanotechnology. Next is the introductions of nanoparticles. Nanoparticles, it is the particles which lie between 1 to 100 nm in the range. Nanowave derives from the nanos, meaning drop or small. So, nanotechnology term coined from was the audio technology in 1974. And nanometer is 1 billionth of a meter. Nanotechnology has the 3 dimensions: 1 dimension, 2 dimension, and 3 dimensions. Okay. In 1 dimensions, any particles lie within the 1 dimensions at the nano scale. In 2 dimensions, diameters or length and height. Minimum 2 sides of the antimaterials lie between the nano scales is called 2 dimensions. And in the 3 dimension, all parameters of the molecules are exist in the 3 dimensions. That is, for the examples, nanoparticles. This is the parameters which you can conclude it, the size of the nanometer, which is the 100, which is the 1/9 tenth inverse 9 nanometers parts of the 1 meters, which there is a number of molecules, but at the nanometers there is a water molecule, glucose, and there are different size of the molecules, and according to the different size of the molecules there is a number of colors showing. This is the some nanoparticles, the diesel exhaust particles, which is non-engineered. It is remains same as the natural product. The fullerene, which is line the diamonds and nanotubes. There is a engineered nanoparticles which are made up by the humans, dendrimers and quantum dots. It is the some examples of the nanoparticles. There are too many types for the synthesis of the nanoparticles. First is a top-down approach, and second is a bottom approach. In the top-down approach, size reductions to nanoscale for assembly and made up a building block. For the examples, whenever a bulk material is cut down into the fine piece and make a scaffold or a statue. It is examples of the top-down approach. In this process, mainly physical process are live. And next is a bottom-up approach. In this approach, chemical and biological process are live. In this small atoms and molecular assembled and make up building blocks or nanoparticles. This is mostly preferred approach for the synthesis of nanoparticles. In this discussion, we will use the bottom-up approach for the synthesis of nanoparticles. There are some properties of the nanoparticles which can reduce the size of the nanoparticles, and there are different size of shape nanoparticles compositions with each other, and have unique for the multi-finger functionality. And surface modifications, there are surface modification for the any surfactants attached, and chemical reactivity increase, and some nanoparticle assembly for the reactivity and used in the most reactive and chemical reactivity size and surface ratio may be increased for the assembling nanoparticles in which used for the catalytic activity of the nanoparticles. Here are some examples for the synthesis of nanoparticles. Green synthesis of palladium nanoparticles from palladium chloride solution using leaf extract of Rosa damascena plant. After the 15 minutes, we observe the color change in the solution. Color of the solution turned from yellow to brown, which indicate the formations of palladium nanoparticles. The appearance of brown color in the solution indicates the reductions of palladium ions to nanoparticles. This is the flow charts for the synthesis of nanoparticles. Plant extracts plus palladium chloride ion mix take a time to the stirring, and palladium nanoparticles in solutions observe. This is the first qualitative analysis, that is the yellow color to brown color, and next confirmation of the palladium nanoparticles is confirmed synthesis, quantitative analysis from the UV visible analysis, and next purification and recovery for the FTIR, TAM and SAM, and next we will check for the biomedical applications. Next, this is the Rosa damascena leaves, and we picked the fresh damascena leaves from the plants of and cut into the fine pieces for 14 days, dry in air. And after that 14 and 15 days, leaves color change green to yellowish, but after 20 days leaves color absolutely change in the brown color. It will be very fine, make a very fine powder, and 10-gram measure of the plant leaves, and heating using the heating mantle for the 20 minutes at the 60 degrees Celsius. After the 20 minutes, we get plant extract, and Bodman filter paper used for the extractions of the plant extract, and after the cooling plant extract, it will be will be mixed in the palladium ions, and after 15 minutes use of the magnetic stirrer, I will get palladium nanoparticles. It is the qualitative analysis, that is the change in the color, which due to the properties, nanoparticle properties, is the after the quantifications is the after the qualitative interpretations, it is the quantitative interpretation for the UV visible analysis in the 360, and I will get the confirmations of palladium nanoparticle has been synthesized. And there is another contributions is the TAM analysis, is the confirm 20 nm nanoparticles found, and after TAM characterized for the nanoparticles IFTIR, there are some peaks which indicate the phytochemicals present in the leaf extract, which convert to the ions into the nanoparticles, which is responsible for the conversion of the nanoparticles. This is the antibacterial activity of the synthesized nanoparticles. There are some functions which lie for the destruct of the antimicrobial bacterial cell wall. There are some nanoparticles, ultra small nanoparticles enter in the cell wall. Positive and negative damage the DNA and protein denaturations, and enter the bacteria, lastly bacteria cell wall. There is an example for the. Still, there is a strand for the Staphylococcus aureus, and this is Escherichia coli, and this is Pseudomonas aeruginosa. This is a table for the find out the antibacterial and nanoparticles susceptibility inhibitions on for the antibacterial activity and antibiotic used for the this is the 4 number zone for the antibiotic levofloxacin antibiotic used for the antibacterial and organism. This image for the antifungal activity nanoparticles also disturb the cell wall of the cell wall of the fungal strands, and they nanoparticles disturb the genetic material of the fungal strands and disturb the cell wall of the fungal strand enter, and the cell lastly cell wall membrane has been damaged and cell wall also damaged. So fungal also removed from the surface. This is the test for the some fungal strand, Candida albicans, Aspergillus niger, and Aspergillus flavors. There is no more than susceptibility for the and fungal strands according to the antibacterial strands. There after the sterilizations 24 hours, we will find the some parameters no measure no more than inhibitions on find before the antifungal strand, and this is no is so more susceptible according to the antibacterial. So synthesized palladium nanoparticles is no more liable for the antifungal strands. This conclude that the biological rate reductions of palladium ions achieved by Rosa Damascena leaf extract are the best result for the with the use of palladium chloride for the synthesis of nanoparticles. The antibacterial antifungal activity of the synthesized palladium nanoparticle was tested against antibacterial as Staphylococcus aureus, Bacteria coli, Pseudomonas aeruginosa, and as well as antifungal activities by strands like Candida albicans, Aspergillus niger, and Aspergillus flavors. The acknowledgement NFPWD UGC New Delhi provide me financial assistance for PhD research. This is my research paper. Thank you for your attention.